Hello, everyone. I'm Michael Bradshaw, and this is my work on the identification of clusters containing future gene to phenotype relations. This is Jane Doe, otherwise known as my gene 2 patient 1930. Now, Jane seemed like a perfectly normal, healthy child until she began walking. Doctors described her walk as gait ataxia. Following that, Jane began having clonic and tonic seizures, after which she also developed pancreatitis and rapidly began losing weight. Later genetic testing revealed that Jane had de novo mutations in the genes SSPO and NBEA. Now, the curious thing is that if you look in resources like OMIM and Orphanet, there are no connections between any of Jane's genotypes and any of her phenotypes. So Jane has what we call a rare and undiagnosed disease. Paradoxically, rare diseases as a whole are not that rare. Somewhere between 25 and 30 million Americans have a rare disease. That equates to about 1 in 10 people. But while we don't know of any direct connections between these pieces of information, that's not to say we know nothing about them. In fact, thanks to the vast number of biological ontologies, we know quite a bit about how these phenotypes are related to each other and how these genes and proteins interact with one another. Now, if we look at Jane's genes and phenotypes in the context of these larger biological ontologies, and then overlay the known connections in OMIM and Orphanet, it's plain to see that there are potential explanations for Jane's genes and phenotypes, just indirect ones. So it's discovering dense regions of these indirect connections within a network like this that we're interested in this work. The way that we've decided to go about identifying these regions is using network clustering methods. So a clustering algorithm, for example, might identify 3 clusters in this region. This orange one in HPO, perhaps this green one in string, but what we really care about in the case of somebody like Jane Doe would be something like this cluster in gold here in the center that spans the phenotypes and the genotypes and connects terms related to somebody like Jane Doe that were previously not known to be connected to each other, suggesting some type of connection that's meaningful that we can then hand to a physician or clinician for hypothesis generation and further investigation. So this is exactly what we'll be doing in this work. We're going to be creating a heterogeneous network, clustering it, identifying meaningful clusters that we then can hand to a physician or clinician for further work. The way we start this is by creating a heterogeneous network of the human phenotype ontology and the string protein-protein interaction network. We connect these 2 sources using annotations of OMIM and Orphanet provided by HPO. We create what this network would have looked like in the years 2021, 2020, and 2019. These previous versions of the graph are going to be useful later on in the development and validation of our method. Next, we cluster this heterogeneous network using 4 different algorithms: greedy modularity, walk trap, infomap, and Cesno, which unlike the others is aware of known type and propagates that information. Now, these 4 algorithms have a tendency to produce a relatively small number of clusters with a relatively large number of members within any cluster. Some of these clusters are as large as 25,000 members, which, if we were to take 25,000 genes and phenotypes and hand it to a physician as containing the potential solution for somebody like Jane Doe, that would not be useful. So, as identified by another study, it's noted that in order to be useful for human manual curation, a cluster would need to be no larger than 100 nodes in size. So to reach this threshold, we introduce another layer of clustering using the Paris hierarchical method. The nice thing is Paris allows you to set an upper bound for cluster size. We then recluster all the previously identified clusters, which results in having many more clusters, all of which are of this smaller, more manageable size. So now we have all these clusters that are potentially manageable, but we need some way of quantifying their usefulness and knowing which ones are good versus which ones are bad. The way that we decide to go about identifying their usefulness is with the task we call edge rediscovery. The way edge rediscovery works is we first identify clusters in the 2019 data, and then we look for edges that are then added in future years, like in 2020. So with this list of future edges added in 2020, we look for cases where both nodes of a future edge co-occur in one of the 2019 clusters. So for example, if this edge in 2020, if both nodes co-occurred in this gold 2019 cluster, we'd call this a rediscovery. However, if something like the second situation occurred where the nodes are split between the gold and the green cluster, this would be a miss, a non-rediscovery. So we create a null model based on this using snowball sampling and 10,000 permutations to create an expectation of how many rediscoveries we'd have. So looking at the log2 ratios plotted here, if there is an equal number of actual rediscoveries and expected number of rediscoveries, that would equal 0. If there are fewer rediscoveries in the actual cluster than there are expected, this value would be negative over here in red. But if the number of observed rediscoveries exceeds expectation, that would be a positive value, those that we have in green. So in actuality, the vast majority of clusters have less than or equal to the number of rediscoveries we'd have expected based on our null model. However, that leaves somewhere between 7 and 19% of clusters exceeding, and usually drastically exceeding, expectation. So we now have a small handful of clusters that are very meaningful and a large number which need to be filtered out. So we need some way of triaging these clusters before presenting them to a clinician for potential further investigation. The way we've decided to go about doing that is by introducing a machine learning model, in particular XGBoost. We trained an XGBoost model to regress a usefulness score about a given cluster. We trained our XGBoost model on biological features such as tissue, disease, and GO enrichment, and cluster topology measurements such as internal degree, the number of triangles, and the gene to HPO ratio within a given cluster. In actuality, in our 2019 clusters, there are 1,898 clusters with a p-value less than 0.0001, or that is to say that the actual cluster we observed had more rediscoveries in it than all 10,000 of the snowballing simulations. So by pulling that same number of clusters out of the group predicted to have the highest score or to be the most significant, we found that the actual and predicted groups overlapped with 1,351 clusters, or that there was about 71% overlap between the clusters that were the most significant and those we predicted to be the most significant. So we're now to the final step where we've created a heterogeneous network. We can cluster it into manageable groups, and we have a fairly accurate way of predicting which ones are going to be useful for future rediscovery that we can then pass on to a clinician for hypothesis generation and further clinical investigation. So we apply this method to Jane Doe and the rest of the MyGene2 patients. In the case of Jane Doe, we found that the gene NBEA and the HPO term for weight loss co-occur in one of our walk trap communities. The interesting thing is that this result is validated by another undiagnosed disease network that had been updated more recently than MyGene2. The Undiagnosed Disease Network out of Harvard Medical School had been updated a year more recently than MyGene2, and the researcher that updated it noted that they'd solved Jane Doe's case, and that in fact it was the gene NBEA that was responsible for all of Jane's phenotypes, which is the same gene identified by one of our clusters. Jane is just one of 912 patients or families on MyGene2. Of these, 115 remain unsolved today, or 115 have no connections between any of their phenotypes and any of their genotypes. But of these 115, 13 are explained by one of our clusters. That's 13 families that have potentially gone years in a diagnostic odyssey that now have a potential answer. As for future work, we have an ongoing collaboration with Children's Hospital of Colorado and their precision diagnostics team. We aim to eventually include this tool in their diagnostic pipelines. And at this time, I'd also like to thank my collaborators and the various people that have been instrumental in this work along the way. Thank you.